Did you ever wonder what the overlap between studying for the MCAT and bodybuilding is? Probably not, but after this lesson, you'll know what it is. My name is Alex. Welcome to this high yield science lesson on hormonal regulation of metabolism. After this lesson, you will be able to predict the mechanism of action of peptide and steroid hormones, and you'll also be able to contrast the anabolic effects of insulin and testosterone. Take a few seconds to read this question stem now. Right off the bat, the link to bodybuilding is pretty obvious, but besides that, the stem highlights some of the key differences between insulin and trenbolone. We're not told much about the nature of insulin, besides that it has a wide variety of anabolic effects. That being said, you are expected to know for test date that insulin is a peptide hormone. Trenbolone is a steroid hormone, as evidenced by its suffix and by the fact that it's able to bind androgen receptors. Same deal, it has a bunch of anabolic properties. The question here is, how can they have the same effects despite acting differently? To answer this, we'll have to consider their mechanisms of action individually and how those lead to their anabolic effects. Let's start with trenbolone. We're told that it's able to go straight through the cellular membrane. What are the properties of compounds that are able to do this? Nonpolar, small, uncharged, hydrophobic. We were able in the question stem to determine that trenbolone was a steroid hormone. Not only are those hydrophobic, but their core is cholesterol, and cholesterol is found in the cell membrane. What role does cholesterol play there? It increases the fluidity of the membrane. If the membrane is made of cholesterol, and like attracts like, or as I like to put it, like likes like, then steroid hormones won't have any problem passing right through the membrane via passive diffusion. Accordingly, the receptors for steroid hormones are found either in the cytoplasm or in the nucleus of cells. Our second prompt here requires a little bit of critical thinking. We're told that trenbolone increases ammonium ion uptake into the muscle. What are the three categories of macromolecules, and which category would this effect be relevant to? The three categories are carbohydrates, lipids, and proteins, and of all three, only proteins necessarily contain nitrogen. We're interested in how ammonium uptake contributes to muscle gain. Well, ammonium is a protein precursor. Again, nitrogen is part of every single amino acid, so ammonium intake would favor protein formation. I think ultimately this is somewhat analogous to Le Chatelier. You have a balance between free ammonium ions and the nitrogen incorporated into amino acids. So more ammonium signals the need to synthesize amino acids to reach the equilibrium again. Okay, this is somewhat of a stretch, I'd say, but it's always good to relate topics, especially across disciplines. Shifting gears to insulin now, we said it's a peptide hormone. How do those differ from steroid hormones in terms of permeability and onset of action? Peptide hormones are gigantic compared to steroid hormones, at least in the micro perspective of things, so they're definitely not able to cross the cell membrane. Not only that, but they contain both hydrophobic and hydrophilic regions, the latter not being conducive to permeability. So we see here, not able to cross the membrane. But because hormones require receptors and must be able to bind to them, the receptors are located on the membrane. So ultimately, peptide hormones exert their effects by generating a cascade inside the cytoplasm, which involves secondary messengers and oftentimes phosphorylation. And that whole cascade happens fairly quickly, which leads us to the point of onset. Peptide hormones have quick onset of action, but usually their actions wear off quickly. That's opposed to steroid hormones, which take longer to act, but whose actions are usually more lasting. A perfect example of this is ADH and aldosterone. If you have a little change in blood osmolarity, what will be released first, ADH or aldosterone? ADH is the peptide hormone, so it will be released by the posterior pituitary first, act quickly to return blood osmolarity within the normal homeostatic ranges, and wear off quickly. But if you have a hemorrhage or just lose a ton of fluid via sweat, then it's not just a little change in osmolarity. You actually have considerably less fluid in you, and that's going to be detected by the body via a lower blood pressure. Aldosterone is going to kick in, take longer to act, but ultimately have more drastic and long-lasting effects. So in any case, back to insulin. What's the role of insulin? What does it promote? Cells need glucose. Glucose is not much use in the blood besides for allowing cells to pick it up. So insulin works by promoting glucose uptake. To be more precise, insulin induces an increase in the uptake of glucose by inducing the translocation of the GLUT4 transporters from vesicles inside the cytoplasm to the plasma membrane. What happens if you have too much insulin? You drastically reduce the amount of glucose in the blood, leaving not much left for the cells to pick up. What important organ primarily feeds on glucose and whose decrease in function might lead to the symptoms in the question prompt? It's the brain. You might have thought about the heart, but actually, The heart can get 100% of its energy from fatty acid oxidation if it needs to, so it's really the brain that's all about glucose. Let's wrap things up with my favorite prompt of this lesson. We're told insulin increases the rate of glucose transport across the cells, which is consistent with what we've just seen. It also decreases the rate of lipolysis and increases the uptake of triglycerides and amino acids from the blood. The really cool thing here is that we pretty much could have inferred all of this. Let's see how. Carbohydrates first. We said insulin increases the transport of glucose into the cells, so cells have more of it. Glucose is so important in the body. Pretty much every tissue feeds off of it, so we don't want to get rid of it. We want to save it. That's why in a normal state, all the glucose is reabsorbed at the proximal convoluted tubule of the nephron, and none of it shows up in the urine. How is glucose stored in the body? It's stored in the liver and in the muscle cells in the form of glycogen. Glycogen is really just a bunch of glucose monomers stuck together with a glycogen core, but really, glycogen equals bunch of glucose monomers stuck together. It's important to recognize that the muscle cannot contribute to keeping the blood glucose within a normal range, since the muscle cannot release glucose into the bloodstream. So when there's a lot of glucose in the blood, for instance after you ate your cake, insulin tells the liver, hey, there's a lot of glucose right now, let's save it up. The liver makes glycogen, and that's called glycogenesis. Glycogenesis. This is not equivalent to glycolysis, which is the breakdown of glucose to two pyruvate molecules. What's the opposite process of that? Breaking down glycogen. And that's called glycogenolysis. In what circumstances would that happen? When the body senses that there's not a lot of circulating glucose. So there's two things happening. The liver is the glucose supplier. By breaking down glycogen, it can release glucose into the bloodstream for other tissues to pick it up. But the muscle can't do that, as we just said. What enzyme does it lack that would enable it to do that? It lacks glucose 6-phosphatase. And the point here is that the muscle isn't the body's powerhouse. Again, it's just looking out for itself by making glycogen, so that if there's ever not enough glucose, it can break down its own glycogen. We said we're making glycogen. It would be counterproductive to break it down at the same time, so that process is reduced. The last thing to consider is gluconeogenesis. What does this process allow to produce? Glucose. And what is glucose produced from in gluconeogenesis? From glucogenic amino acids, from glycerol, from pyruvate. If our cells are being fed glucose, they do not need to go out of their way to make it. So gluconeogenesis would also be reduced under such conditions. Before we look at lipids, quick question for you guys. Why does gluconeogenesis exist if we can get our glucose from glycogenolysis? Because glycogen stores are finite. They last 12 to 24 hours after the last meal, after which the body needs to make glucose from scratch rather than getting it from its storages. Let's look at lipids now. What does breaking down lipids provide? A whole bunch of energy, right, via the form of fatty acids. Why don't we need this energy under these circumstances? Because the cells have a lot of glucose, which they preferentially use as their energy source. You can think about it this way. Is your body going to start breaking down fat after you've just had yourself a cake and ate it too? Unfortunately not. Finally, we have our proteins. We need energy to make proteins, right? Transcription and translation are super energy costly. But at the same time, proteins and protein synthesis are essential for life. So under circumstances, will protein synthesis be upregulated? Sure. We have all this energy thanks to glucose, and that's in fact one of the biggest anabolic effects of insulin. Together, we've looked at the mechanisms of action of peptide and steroid hormones, especially considering their structure, and we also contrasted the anabolic effects of insulin and testosterone. A lot of what we did during this lesson is inference. We inferred which biochemical processes would be upregulated or downregulated based on certain hormone actions. I highly encourage you to keep doing such inferences as you go forward with your practice, but also to see how all of these biochemical processes fit within the big picture of homeostasis. Because once you're able to see the big picture, it becomes much easier to see how the little processes fit within it. Good luck going onward with your MCAT prep.